Good evening, everyone, and welcome. My name is Natalie Hoffman, one of the directors on the Woodsworth College Alumni Association board. Tonight's event is a hybrid event. For those of you on Zoom, please note that we have enabled live captioning. You could view the captions by selecting the CC button along the bar at the bottom of the screen. Also, we would like to inform you that this event is recorded and only the speaker and presentation are being recorded on Zoom. To get started, we commence by acknowledging the land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to live, work, and gather on this land. It is our delight and honor that you are joining us for the final Alumni Café event of this academic year. The Alumni Café series presents expert-led discussions on a wide range of topics that are of interest to alumni, students, and friends of Woodsworth College. Tonight's event is extra special as we are continuing to celebrate Woodsworth College's 50th anniversary this year, and for the first time, we present our very own Woodsworth alumna. On the agenda for tonight, we will hear a lecture from Professor Dockstader titled Career Ready, Skills Training and Active Learning in Undergraduate Life Sciences. A Q&A session will follow the lecture. For our audience attending online, please submit your questions in the Q&A tab of the Zoom, and we will include them. I'm excited to present tonight's speaker. Professor Dockstader is a Woodsworth alum who completed her Bachelor of Science from U of T's Department of Psychology, then her PhD in the Department of Medical Biophysics, and a postdoctoral fellowship and research associate position at the Hospital for Sick Children. During her time in the pediatric brain tumor program, she used neuroimaging to investigate how brain structure and function underlie cognition and behavior. In 2015, she left Sick Kids to join the University of Toronto as an assistant professor teaching STREAM in the Human Biology Department, or HMB. In 2023, she became an associate professor teaching STREAM. Her training has provided her with a breadth of research and training experience. Her goal is to create hands-on undergraduate training so that students feel confident and competent in their technical and transferable skills. She has developed courses such as clinical neuroimaging and extracurricular opportunities, such as the HMB Lab Bootcamp, that directly provide students with the training needed to enter the workforce or continue their education. We are honored to have you, and we are eager to hear your lecture on such an interesting topic. So I will hand it over to you. Thank you.
So my name is Colleen Dockstader, and I am a Woodsworth alumna as well. Are you all Woodsworth alumna in the room? Yes. And I wish I knew online, but I assume some people are online as well. That's super cool. I did not realize until it was actually in the news that it was Woodsworth's 50th anniversary this year. Do you want to guess how many years ago I graduated from Woodsworth? What would be the polite answer? So I think I have it on the slide, so don't worry. But I started here in 1993, and so that would have been like 30-ish years ago. I don't think I realized at that time that Woodsworth would have been relatively new. So that's super cool. Is it the newest college? There's not one since then, right? Very cool. I love it. Okay, and so I will go through things a little bit more. I'll talk about. I think I have an agenda for this. But I will talk a little bit about my background, what I did 30-ish years ago and what brought me to now, what I actually do now. Can you hear this okay? What I actually do now. And this is my second career. Third if you include bartending, but I took that off. But I did it 10 years. I count that as a career for me. But I think I took it off the slides for today. So this is my second career. My first one was in research, and now I'm teaching STREAM here at U of T. So I'm going to talk about my path, and then I'll talk about teaching at U of T. And I'm going to just focus on some things that I think are really important, and so that goes to the title of the talk, is really about preparing students for leaving school and going out and getting jobs or creating careers for themselves. And one of the things that I noticed strongly when I became the teacher rather than the student is that you do, as a student, so much of your learning by just sitting there listening to me, right? It never became so, I was never so aware of the format of learning as to when I started teaching, and I realized everyone's sitting there with their laptops out, and all I could hear is I'd say something, and then you'd hear, and then I'd say something else, and then if I say something and they didn't type, or how much they typed, or the looks on their faces, but it was like I created this, you create this communication with your students that's very much just listen to me. Just figure it all out by listening to me. And I was talking about what I felt was complicated stuff. And so having a background in research really led to some of the choices that I made in my teaching and how I wanted to instill skills in students beyond getting really good at taking notes and memorizing. Because I found there was too much taking notes and memorizing. And I probably did that too as an undergrad, although it was like this. Did anyone else have to take notes with pen? Does anyone take notes with pen and pencil now? Anybody? Yes. Good job. You know, actually, it's better for your brain too. Did you know that? That's a neuroscience thing. It's actually better for you to write. I tell my neuroscience students that. They don't care. Type, 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 type. Or on their phone, thumb, 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 thumb. So I'm going to talk about all of this and within a specific context. So how do we create competence in a field so that when students graduate, they feel like they know enough to go do something? And it does not have to be, although it tends to be very often in the field in which I teach. Very often these students want to go into medicine or they want to go to grad school. Of that, I don't really know how many do. I'd certainly say less than half, but I'd say more than half, certainly that's what they would like to do. And so there has to be other choices than just that. There can't be just those choices. Even though those are awesome choices, there are many more choices. I won't talk about the different choices today. But if we can cultivate a degree of feeling competent in your field, the students might be a little bit more comfortable in foraying into something that's different, something that's not maybe the traditional route. And one of the things that I've learned on my journey since I started teaching is competence in a skill isn't enough. It's really about developing the confidence. And what I noticed about U of T students is that U of T students that I work with are often not confident about themselves. They're super stressed, they're very anxious, they're super hard on themselves, and they don't feel like they have the skills or the knowledge. There's a whole bunch of psychology and all sorts of other things that go into that, and we're not going to talk about that as well. But I want to talk about cultivating competence and confidence, like feeling good about yourself in terms of what you've learned and how you can apply that. And so then comes the third part, like how can we do this? And I'm not going to talk about every degree and everything. I'm going to focus on what I know, so I'm going to talk about research. I'm going to talk about research as an undergrad, so how do we cultivate skills within a field so that if someone wants to go into research, and that can be graduate school, you can do research through med school too, or a lot of people tend to want to do grad school, maybe a master's and then med school, but you can also go into the research field immediately following your undergrad with the skills that you've got. And so how do we adequately prepare them? And it's not by having them sit in the chairs and listening to me and taking notes. That has to happen, but it's not enough by itself. And so the agenda is really, I'll just talk a little bit about my career path and my research. And I'm going to give you 2 examples of what I've done since I've come to U of T to try and cultivate skills that go right into that person's CV. Okay? I even tell them how to put it into the CV, how to word it, explain the entire breakdown of that. We create certificates and all sorts of things for these students. And so 2 examples. One's called HMB, that stands for Human Biology, which is the program I work with, Lab Bootcamp, and then a course that I created called Clinical Neuroimaging. They do completely different things at different ends of the life science spectrum, but I'll give you examples from both. And I honestly don't really have more of a slide about competence and confidence at the end of it all, except for some takeaways from everything else I present to you. And if we have time, we can have a discussion around that. So how I got here, this might be absolutely too small to see for those of you in the room, and it's okay, because I'll just read through it, and I'll just read through it anyway. So essentially, my point is in showing this, and I've shown this before, and this is really important to students. Is anyone an undergraduate student right now? Not in the room. No? How about online? Anybody an undergraduate student online? You just have to put your hand up or say yes in the chat or something like that. No? Anyone? No one. You're all done. You can all tick that box. Okay. All right. So I started my undergrad in '93. I was a mature student. That's where the bartending career came from before that. I was a mature student, and I'm so grateful for that. And I'm so grateful for Woodsworth. Thank you, Woodsworth, for believing in me, even though I had a somewhat less direct route from high school. So thank you for that. I did school part-time, and because I had to work, I had to keep working through it. I had to pay my bills. And then once I figured out what I wanted, I just kept doing classes. I did them. I was full-time in the summer and all the way through. At the end of it, I thought, I'll get a master's, but not because I wanted to go to grad school or do research. It's not a great, I think a lot of people might choose to do masters because they're not sure what to do. And I was one of those. And that's okay, because what it did is it gave me experiences that allowed me to go, oh, I love this. And I'll tell you about some of the ones that I thought, oh, I don't really like this. And so by creating different experiences, or by providing myself with different experiences, hence my crooked path, right? You try different things. You figure out what you like. And so when I went into grad school, I was with Department of Anatomy and Cell Biology. Natalie introduced me as medical biophysics, because there is no more Department of Anatomy and Cell Biology. I was the very last person to graduate from that in 2003. And then I went and did a postdoc at Sick Kids. From there, I got a research position, and I worked in research in the pediatric brain tumor program. In 2015, I decided to completely switch gears and go from research to teaching. And in a nutshell, that was very much actually motivated from personal influences that were happening in my life, actually outside of science. I became a single parent, and I had 2 kids, and I was trying to figure out how to do it all and how to be good at everything. And for me, the decision was, I don't even know if we should record this, but whatever, was I needed good benefits, I needed a pension, I needed stability, and if I had to keep signing contracts at Sick Kids one day and not know who else pays my mortgage if they don't renew my contract, it was too much. So that was that. So I applied for a position here, and I got it, and I was very grateful for that. And it turns out that this is the best career for me. And I kind of wish I'd done it earlier, but if I'd done it earlier, I wouldn't actually probably be here at least talking about this, because everything that I did in my research career up to that point really informed how I teach. So via this crooked path and the different experiences I gave myself, I got really lucky, which didn't look like luck at the time, because I ended up with a background in my undergrad and my graduate work in basic and translational research. And I'll explain what that is in a moment. I'm talking about life sciences today, and I'm going to talk sciencey, but I'm not going to use jargon. I hope I don't. And if you don't understand something, please ask. And you don't need to know science to understand it. So I have experience in basic and translational research, and I actually really loved all sorts of aspects about that, but I didn't love what I was actually doing. The research questions that I was asking and what I was doing was actually not for me. So when I went and did a postdoc, which is postdoctoral research after my graduate work, I chose something in a clinical field, and that's very different, and I'll get into what the differences are in a minute. And so I started doing clinical research. I was very lucky to do that. I had a bunch of people say, don't go off and do a postdoc in something completely different, because now you're like this hodgepodge. You've got basic and you've got all this animal research, and then you go off and you work with humans, and it's too different, and what does it say at the end? Well, what it said at the end was really good if I wanted to do a teaching stream position, because it said that I knew both the bench and the bedside. And so in this case, it actually was really informative and helpful for me. And then from there, I came here after I got the heebie-jeebies of constantly signing these one-year contracts, and came to the human biology program, where I got a position in neuroscience. I was as assistant professor, and I got my associate professor, which was my long 10-year plan, last June, 2023. So I can start to relax at 54 years of age. That's why you take a break when you can take a break. Natalie just got her PhD like last month. She's going to take a break. Good girl. Okay. So this eclectic background was really helpful for me when it came to teaching, because when it came to teaching, now I had skills actually in what we call basic research as well as translational research, and that comes from my undergrad and then mostly my graduate work. And so just so you understand what I mean when I say that, basic research are things that will, it can be knowledge for knowledge's sake. You just want to know how it works, which is a super cool thing to do. But it doesn't have to contribute to understanding a mechanism to support someone who has a particular challenge or disease. You can use mice, you can use animals, you can use worms. What do you use in CSB, Natalie? You can use plants, and you can use computers. And these can ask fundamental questions so that we can generate new ideas and principles and further our scientific knowledge. I also got to do translational research, and that was kind of like the bread and butter of what I did, and it turns out translational research wasn't really for me. I really loved what I was doing in a general way, but I didn't love what I did on the daily. So translational work is generally working with animal models. It's not working with humans, and translational work is creating maybe an animal model in which to model a human behavior or condition or disease. And so I was in the field of addiction, and I got my little mice addicted to cocaine or opiates or ethanol. I would put them into withdrawal. I would measure their behaviors. I would look at, what I was doing was looking at brain circuitry and the genes that governed certain things. This is all translational research, because that information informs, albeit mice and not people, but it informs the types of mechanisms and systems that underlie addiction in people. And so the type of work that I did, essentially, I got a few papers out of this. I worked with animals that were mostly, not always, but mostly I worked with what are called knockout mice. So these are mice that one gene isn't working properly, okay? And I would create these little mice addicts, and I would put them in environments that they associated with either good feelings or bad feelings. And in that case, the good feeling was the reward of the drug, the I like it part, and the bad would be the withdrawal of the drug when they don't like it. And my whole point was really to look at the genes. So these would be knockout mice that had this gene that was mutated, to look at the genes involved in the circuitry in the brain, and under which conditions these genes or the expression of these genes seem to modulate certain behaviors. Okay? So I won't give you a whole talk on that, but this is the type of work I was doing. And so I would slice up, after the mice had gone through their experiments, I would give them a lethal injection. I would remove their brains. I would slice them up. I would stain them. I had performed surgery on them. And always I was asking these fundamental questions that were translatable to applying them to understanding the human brain and addiction. And so one paper that I did a bajillion years ago is this one, and it essentially was looking at the role of something called the D2 receptor. You've heard of dopamine. I bet you everyone's heard of dopamine. Dopamine tends to be the I like it chemical. Okay? It has to work by binding to little receptors that are sitting on cells, and a receptor sitting on a cell is like a lock and a door. And then dopamine comes along, and dopamine's the key to turn the lock. And then off it goes into the cell, okay? And so I would look at these locks, and my lock would be the D2 lock. And dopamine, if it bound to it, would it cause someone to say, I like it? In this case, my mouse. And so what I found is that sometimes the D2 receptor was really important, and other times it actually didn't help at all, and that really depended on the condition that the animals were in, whether they were drug naive or they were addicts. And so that led to information that told us that certain locks for the dopamine chemical were really important in a naive, non-addicted animal, but not an addicted animal, which can inform treatment and relapse and all sorts of things. Now, in and of itself, it doesn't, obviously, but hundreds of publications that show the similar findings can really start to move the world towards clinical trials, which then is working with humans. And so this is the kind of work that I did, but I didn't like doing what I was doing to the mice. It wasn't for me. I'm a vegetarian. I was back then. It wasn't for me. And I was doing okay until I had my first child. I had my first child while I was still in grad school. My second one, I was in my postdoc. And I was doing okay until I came back from mat leave. And then every time I had to inject these little mice, I just started crying. Every single time. I don't know if I thought they were my kids or it was my hormones, but after that, I was done. So I appreciate the research. I understand the reason for it. I'm not advocating for or against it. I just knew at that point that that wasn't for me. But I loved research. I loved asking questions. I loved figuring things out. I loved being the first person. I made it bigger than it was. But I always felt like when I'm putting the data in and pressing enter, and you're about to get the statistical result, I always thought, I'm the first person in the world to find out this result. Not that anyone else is asking the question necessarily, but I get to be the first person in the world to know this. And I loved so much of the research process. I absolutely wanted to stay in it, but I wanted to work with people. I wanted to work with the people I was trying to help, not indirectly, but directly. So I got really lucky, and I found someone at Sick Kids who believed the argument that I said, I will be a better clinical researcher by being informed by my animal research, this more basic and translational research. I'll have a different perspective on it than someone who's maybe always been on the clinical side. So she said, yep, okay, I'm up for that. So I got into a lab at Sick Kids for my postdoc with Rosemary Tannick and Doug Shane. Rosemary Tannick just retired from OISE, actually, and Doug Shane's still working. And I learned how to do neuroimaging, which to me was something that's always been super cool. And so this is looking at brain structure and function of human brains. Clinical research is always working with people. As soon as I say clinical research, I mean people. If it was a vet, they would mean animals, but anything else, we mean people. It can still be basic. We can still have basic clinical knowledge, like learning for understanding's sake. But a lot of clinical research, the vast majority, is going to focus on something that's related to understanding, diagnosing, creating prognoses, understanding disease, and how to fight that disease. And a lot of the clinical research that you're probably most familiar with would be when you hear about clinical trials. Okay, clinical trials is when that translational work is ready to move to humans. So I did clinical research for many years, and I'm so grateful for that. So I did 12 years of clinical research, also called bedside research. You don't need to be able to read or understand anything on this slide. It's a hodgepodge of some of my publications that came out. It's a demonstration or a tiny little snapshot of an MRI. This is not from Sick Kids. This little guy came from the internet. We don't have pictures of our patients that we post online. This is our MEG. This is an older person who was 18-year-old who was put into the MEG, and this is our MEG. MEG stands for magnetoencephalography. Oh my God, I can't say it. MEG, magnetoencephalography. And this was really my bread and butter. And between using the MRI and the MEG, we were able to look at brain structure, look at brain function in terms of time of activation, location of activation, and this little guy here is called a time frequency response and is basically telling me how fast the cells in the brain are firing during these responses that you might, here we go, which is the coolest thing on the planet, to me. Is the coolest thing, that you can look in someone's brain and see where the activity is, how strong it is, and how fast those groups of neurons are firing. And that becomes a very important question when you start working with different populations. So I worked with kids who had ADHD, I worked with kids who had OCD, and I worked with kids later who had brain tumors. And all of them were malignant, and I thought I would cry the entire time. You thought I'd cry with my mice. It was very hard to work with kids who have brain cancer, obviously, but also so rewarding, so incredibly rewarding. And my job was really to understand, not the cancer actually, my job was to understand how radiation, which was a treatment for every single one of these kids, because if they didn't get the radiation, they'd pass away. So there's no choice at all. The radiation destroys more than just the cancer, right? Unfortunately, no matter what you do. And that's a big lecture all by itself. And so of these children, about 80% of them had cognitive deficits to a point where about 80% of them were not able to get into college or university. And so my job was really to understand actually the damage post-treatment to these children who had all survived, to understand what radiation is actually doing to the brain that causes these cognitive deficits. And so that becomes basic knowledge, but as we disseminate that knowledge, other people can use that to inform. And so we were able to start, the idea is, not with just my research, but hundreds of people doing the same thing. We can start to customize research. Then we get into personalized medicine, where people are given a treatment approach that's best for them, and it's not a prescription that's generic. And so something like that might look like this. And this was a publication that came out, and obviously, well, I don't know if this is obvious, sorry. This is someone's brain. Was everyone knew that? Okay, sorry. Okay, so this is someone's brain. This is actually the back of their brain being active when they see something. So this is the visual cortex saying, I see something. This is a timeline that says when they see it, how strong the activation in that part of the brain is. And here's my favorite picture, which is a time frequency response that says how fast groups of brain cells are firing together in synchrony, so at the same time, during the I see it phase. And what you can see here is just use your own, just look at it like you would two pictures and ask yourself what's different between these two pictures. And here we have the control group, and here we have the patient group. And you don't need to know anything about science to be able to see that there's differences. So it'd be my job to interpret what those differences are and what that means, okay? So I loved this. I absolutely love doing this. Even when I left Sick Kids, I've continued my collaborations where I can because I love working with patient groups. My favorite paper that I ever did, I actually didn't do any of the data collection or meet the patient. It came from this little guy named Zion who was at the Children's Hospital of Philadelphia. When he was 2 years old, he lost his hands and feet were all amputated because he had sepsis. He had an infection that ran through his entire body. Your limbs get hit the hardest, and they had to remove his hands and his feet. When he was, oh gosh, I'm making it up now, like 7, 8 years old, he got a transplant. Maybe 6 years old? This is my brain. I don't remember things when I wing it. He got transplants for his hands, which was amazing. He was the first pediatric bilateral hand transplant on the planet, which was super cool. These are his new hands. They came from another little person who passed away, and the family donated their hands to Zion. So these are Zion's hands. There's many publications that have come out because he was the first little guy. The most recent that came out that I had the privilege of being on was actually just last summer. that we were able to publish this. And this was just a longitudinal, meaning long-term study. So we wanted to see 6 years later, what is his brain activity like? How does that compare to the performance that he has? Has it normalized to a similar 14-year-old kid or 12-year-old kid? And so without, again, having to get through the science, he's getting little puffs of air to his fingers. They're showing responses, where those responses are located, the strength of those responses, and how they might compare to someone who is of a typical age. So essentially, Zion can feel everything. He can write, he can throw balls, he has dexterity, he has gross motor skills, he has fine motor skills. His quality of life is exceptional relative to what it was in his words. His activity is super high compared to someone else's 12-year-old boy doing the same thing. There's lots of interpretations for that. That's not part of this talk as well. So, but these are the cool things that I've had the privilege to work on over my time. I've also had the privilege of working with fourth-year undergraduate students. So when I came here, I didn't want to lose the research. So I took on undergraduate students who are in their fourth year. And so these are called thesis project courses that we offer in the human biology program. And I was able to use data that I was collaborating on at Sick Kids. And these kids spent all their time in front of a computer learning how to do those fancy neuroimaging software techniques so that they could figure out structure and function. And this was a group of children who had OCD that they worked with, so obsessive compulsive disorder. And they characterized things like location of responses, density of gray matter in their brain, the pathways, the strength and thickness of the pathways that transmitted the information, all sorts of things. They did awesome jobs. How much more time do I have? About 13 minutes, right? Okay. So now, that's my life, up until now. So now I come to U of T, and I want to continue my research, so I work with the fourth-year undergrads, and that's great. And I come and I do my lectures, and I realize everyone's actually, they're taking notes. Everyone's taking notes, and I realize, which is really interesting, that I was giving a lot of case study questions on exams, and my students lost their minds. My first year of teaching, I realized the wrong way, because my students were my guinea pigs, I guess, that they had no idea how to do a case study. They had no idea how to answer a case study. And it took me a while to really dig into it. It was like the next year, I'm like, okay, I'm going to give you a case study. This is what a case study is. We go through some case studies. But then I give a case study for the final exam, and people blank and don't do well again. And I thought, what is going on? And so U of T is a big place. Your classroom sizes for first year are thousands, literally. Second year, hundreds. Third year could still be 100. Fourth year, I teach 100, one class that has 147, one class that has 201, and then a couple seminar courses that have like 30. So they're used to tests that you have to memorize a whole bunch of material for, and then you just kind of go blah onto the page, be it multiple choice or short answer or something. That's what they're used to, because when you have hundreds to mark, or dozens to mark, or thousands to mark in first year, what can you do besides the Scantron, right? So it took me a while to realize that a lot of students would use the technique of memorizing things. And that's what they do when they'd sit. They'd write down what they thought that was the most important information, and they read it, and read it, and read it, and read it, and then put it on the paper. And then how many remembered what they wrote after that, like 2 weeks later? Not so many, right? And what is the application value of what you learned? There's lots of value collectively. You do this for lots of courses, you still gain a ton of knowledge, obviously. But it's different when you're learning something conceptually than when you're applying it, right? And even when we apply it, which is amazing because we have so many wonderful labs, and the students are able to access the labs, The labs have thousands of people, so they have to go through their rotations, and you get 2 to 3 hours, and everything's very prescribed for you. It's like you were to bake a cake, right? They'll tell you do this, then do this, then do this, then do this, and then you'll have a successful output, and I'll go right up about that. And all of that is okay. It's my background, it's many people's background, and it's a great place to start. But what if we could give them a little bit more? What if we could give one more layer? And that's the experiential learning. And I'm not inventing anything new. Obviously, people have been doing this forever. There's experiential committees at U of T. It's developing all over the place. So I'm not suggesting that this is something I started, but it is something that I felt was super important to bring to the human biology program, which is an undergraduate program. It has like 3,000 students in it. It's the largest undergraduate, I think, the largest undergraduate program in Faculty of Arts and Science, I think. You can please correct me if I'm wrong. So at the time, the director, my boss in my program, was Melanie Wooden. Anyone know who she is? She's the Dean of Arts and Science now, which is really crazy cool, and she's a really lovely person. And we are very lucky to have her. And she and I were like, let's bring research to the students. And so we wrote a grant. I thought I'd never write a grant after I left research. What's the thing she said to me when she's like, you know how to write grants, let's write a grant. So she and I wrote a grant, and we got almost like $150,000, a little over $150,000 to try an experiential learning program with the students. And we called it Human Biology Boot Camp. And I called it boot camp because I was really into exercise at the time, and I wanted students to think, this is hardcore. You are not getting out of this without like breaking a sweat. And so in a nutshell, these are some of the labs in the human biology program that we have. We are so lucky to have amazing labs. In a nutshell, what we did is we took on 48 students. The 48 students were broken into groups of 4. All of them are 3rd or 4th year students. Some of them are 5th year students. Each of those groups of 4 were assigned to a facilitator. A facilitator is not a TA because it's a different position, but many of them, it's very, very similar. They'd be either graduate students or postdocs. We were lucky enough to get a lot of postdocs who did this. And it was an experiential program that allowed the students to get introduced to what life was like in the lab on a regular basis. Not come in and learn a skill in 3 hours and leave because we gave you a recipe, but come in for 2 weeks from 9 to 5, which is actually the only thing they really didn't like. Please, do we have to be in here at 9? Yes. Please, do we have to stay to 5? Yes, we did. But you get a 1 hour break for lunch, and anytime you need to go and do something, do something. But we made it like regular, old school, every day, here we go, 8 hours, let's go. And but there was no homework, so that was good for them. So it was 2 weeks of being in a lab and working in experiments in a way in which the students had to do a lot of their own self-teaching. There was no memorizing to be done at all. We gave them a problem. It's okay, I'll read it out loud, but it is totally okay if you don't understand what the problem is. I barely do. I had to hire someone to develop the protocol because this is not my background. This was a molecular biology type lab. And so the problem was design, create, and test a plasmid transfection of a chloride channel. Apply this to a mouse model of Huntington's disease. Essentially what they're doing in the first bit is they're creating something using their wet lab techniques, and they're really honing in on their wet lab skills. The second week, once they developed those skills, they would then apply it to a mouse model in which they were actually missing a particular gene, or the gene wasn't working properly. So they got to have this translational component to it. We wanted students who didn't know what they wanted to do. We wanted students who hadn't had much research experience. I'm glad there are no undergrads here actually at the moment, because I don't want them to know how to get in I don't want them to know the secret to our application process. So the secret for me was really, I want people who don't know what they want to do and haven't had very much experience. because the more experience they had and the longer their CV, and the less they really needed this. I wanted people who really needed this. However, you have to have some students who are strong. When you combine 25% strong students and 75%, I have no idea what I'm doing, it actually works amazingly well. And so we've been running this for 5 years now, so we've done 240 students. We have a manager, we have technicians, we have 6 facilitators. We have 6 facilitators, so we have 12 groups of 4 students. Each facilitator gets 2 groups of students. By the end of week 1, they've learned all the skills that they need to execute what they're doing over the following week. So they've learned all sorts of, and this is just lists some of their basic skills. These skills are really important because we selected protocol that would be highly transferable to many wet lab settings. That means if they wanted to go do research at a bench somewhere else, we have given them many of the skill sets that are involved, okay? Not the nuances of all of them and not all the particular protocols, but we've set them with the fundamental skills that they could go to a lab and say, yeah, I feel confident in my pipetting skills and that sort of thing. We also gave them workshops in biosafety, communication, data analysis. By the end of week 2, they were able to take what they learned, the steep learning curve in week 1, and apply it to this translational model. We also gave them workshop in ethics, career preparation, etc. They loved it. They really, really loved it. You have to go to see it. The first couple days, no one really seems to like it. You can't tell. Everyone's quiet. Nobody talks to each other. Everyone's scared of doing mistakes. I'm walking around saying, you got to do up your lab coat, no ankles, roll down your pants, because of splashes of equipment and stuff like that. So everyone's kind of really nervous. But honestly, the love that happens after all of that is absolutely amazing. A typical day would be to review what they're going to do. They might have a workshop. When they do have a workshop, we bring in the big guns. We don't do it ourselves. Our research ethics talks are given by a professor at Atlanta who is very head in bio research ethics. They do bench work. They discuss it. They have a lunch, which they get very excited about. And we do a journal club. We do ask them to read something and go through it, and they might do some more bench work. And at the end of each day, the students will lead the discussion about what worked and what didn't work. These are real pictures from our labs. We have really amazing equipment. The students get very, very lucky. They get a manual that gives them a lot of the information, but it doesn't tell them exactly what to do. It's like giving them a recipe for a cake and telling them what the ingredients are, but leaving out how much for each. Or telling them most of the ingredients, but leaving out some of them and just putting blank. And their job really is to go and figure out how to define that protocol exactly, because we've given it to them generally. And so we prompt them with questions like, what are the major steps? What are characteristics of? Which one should you use? Why? That sort of thing. And the students will go off into groups, and they'll go to the coffee shop, or they'll go down the hall to the lounge, and they will get on their computers, and they'll figure it out, and they'll figure out what's missing and what is the right recipe. And then they meet back with the facilitators who will correct them, but correct them in a way that doesn't tell them the answer, but says, this is great, you're exactly right. This is okay, but what would happen if? And then they guide them through. And the facilitators are amazing at bringing these students to a point where they understand which corrections need to be made to their calculations and why. They also do a lot of online research to look up papers online. They spend a lot of time doing work together. They spend a lot of time learning how to do presentations. And I don't mean fancy presentations. If this is fancy, I'm not sure. But I don't mean like rehearsed presentations with proper slides. I really mean like they would just learn how, at the end of the day, to put a couple of pictures up on the screen, show it to their lab mates, and have discussions about their work. And it went really well, even from the first year. You know, you think that you make mistakes and you have to figure things out, and we did, but they were mostly actually about the protocol. Like, we decided partway through that, you know, this protocol wasn't ideal for certain reasons, and so we might want to change things. But in terms of the student experience, it has worked for all 5 years. We are not changing anything. And I have been asked by FAS if we will change things a couple times, because the biggest thing is that it's free. It's free to all the students. It comes after their classes are done in April and before their classes start in May, although this year, unfortunately, the timetable didn't get any time off between exams and then when summer courses start. So people have to make a choice between this and their summer course. But it's free. 75% of the students have not had research experience aside from what's happened in the lab, and most of them don't know what they want to do. And the whole point is to provide them a deep dive into this is what it would be like to work in a lab. Whether that is as a research assistant, a technician, maybe more of a mentor, a supervisor, this is what it would be like. And so at the end of this, every year, so for the 5 years, it's going to be our 6th year coming up, so we have 240 students who have so far responded. And when we ask them, what was your boot camp experience like? And they say how rewarding, unique, or stimulating it is. Between extremely and very is pretty much everybody. They really find it a very different experience that they really, really enjoy and only have great things to say about. The next thing, we'll just look at the colors here, before and after. How would you rate your confidence with respect to different skills, okay? Before the boot camp and after. And it's the same on the left and the right. And all I really want you to do, although my layout was terrible, is note the colors change from being primarily, you know, gray, orange, blue to gray, yellow, green. And what that means is they go from saying, I'm not at all, slightly or moderately confident, to most of them saying, I am moderately, very confident, or extremely confident. That's a lot to say for 2 weeks' work, but good for them for saying that. I suggest on their resume, rather than saying extremely confident, you say, what did I used to say? But I said, like, don't push it, because next thing you know, they're going to be saying, well, why don't you just start cloning something for me right now? So you say, very comfortable with. Anyway, they were confident, which is the point here. All of them were. Then we followed up with a lot of them. I don't have an exact number, and of course it changes every year, and we can't always get a hold of everyone because they're graduating. But I do ask them, did the boot camp help you? Do you think it helped you secure any future activities? Again, not thinking it's some like magic pill that's going to change their lives. But did it help them? Do they feel that it helped them within the next like 4 months? We usually do the follow-up in September, October, and it happens in May. Do you think it helped you get any opportunities over the summer or for the fall? And the people who say yes, say about almost half of them end up getting an undergraduate lab position. Some of them get a 4th-year project. Some of them are going to grad school. Some of them went into the workforce. And so we say to them, of those who feel it was helpful, what was it that you feel helped you? And they said it was giving permission to make mistakes, being mentored, working with someone directly, having their own independence in doing the work, so we're not giving them the recipe, and being able to practice a series of very transferable skills. So there's all sorts of things that I didn't talk about that happen in the workshop, like statistics and communication and things like that. And this is the biggest one, without assessment. Nobody ever gets to experience that. The undergrads are always trying to get their A's, right? They're so caught up in getting their A's. And so when it becomes this free program that's as equitable as it can be in that way, I mean, they have to take time off work, obviously, but when it becomes a free program that emphasizes supporting people who lack the skills, or at least the confidence in feeling like they had those skills, and then you have no assessment. You will get your certification. You have to come for the 2 weeks. 9 to 5, you're stuck here. But as long as you do the work, you get the certification. And they don't even care about the certification, honestly. They have to email me and ask for it, and half the time nobody does. They just wanted the experience and to be able to put it onto their CV or resume. And without assessment is everything, because they learn that they are allowed to make mistakes. And I could do a whole talk on just the importance of letting the students make mistakes. It changes everything. And so what we found more than anything when we surveyed these students is that they felt more confident. And they felt more confident in their research skills because clearly we'd given them the technical training and they had that hands-on experience. But their confidence increased so much because of this intersection of having that experience, working with people who are mentoring them, working with their peers, and group work is not painful anymore, right? Everyone's doing something and they're not competing against each other, and they all have different strengths, and you can draw on that, and they're allowed to make mistakes. So I'm going to do like a 5-minute snapshot on the neuroimaging piece, and then I have to stop talking. And that's fine. This just says neuroimaging is important. It just says that Canada ranks actually number 2 in the world in terms of output for neuroimaging papers. You can see that in Toronto alone, I can't even count how many there are here. There's a lot of neuroimaging facilities. And U of T is associated with all of these. There's even other ones on top of that that U of T is not associated with, like the private ones. But neuroimaging requires a very specific skill set that nobody, as far as I know, teaches in undergrad in all of Canada. Maybe even anywhere, because I tried desperately to find people in other countries, and all I could find was med schools and grad schools. And so coming from a neuroimaging background, I really thought, this is really important. If a student has any idea on how to analyze neuroimaging data when they leave their undergrad, they're already more hireable in some ways than someone else would be. And so we trained students who took this course called JHA 410 Clinical Neuroimaging. We had them go into the computer labs, and they did 8 weeks of labs learning how to use FSL software, which is the most popular software for neuroimaging in the entire world. No disrespect to Free Surfer, which is the next most. And Brainstorm software, which is a very specific software developed by McGill. It's less popular, but its pipeline is identical to what other neuroimaging would use. This is a product of what my students' assignment outputs would look like. I can promise you, honestly, you don't have to understand any of this, but this is a brain. We're looking down, left, right, front, back. This is a brain, but not any person's brain. So this is just what we call a topoplot, which is like a computerized rendering of a space rather than the brain. This is obviously a brain again. I think it's obvious. In all these cases, the students are looking at either activation or timeline and strength, location, pathways involved. And I promise you, these images and the captions that are underneath them that you don't need to be able to read are publication level. They're totally publication level. This is the kind of things that the students are saying. It's totally publication level. It's amazing. And so I say to my JHA 410 students, and it's been going on for about 5 years as well, has this course helped you procure any opportunities? And if they have, please explain. I know less about how many it's helped procure because I don't know when they graduate. I'm not able to catch up to them all. But I do know that it's helped some. I know that we've had students leave and go to grad school, go to a summer research position, or get an RA, research assistant. They would never get an RA position ever without having any technical experience. And they've been able to do that, and they've worked with Sunnybrook, Sick Kids, Baycrest, and UHN. They've all got positions from. And I say, what do you think the key is to your success? And they say it's working with the same software and the type of data that I would be in the people who are hiring, and using freely available software. And it comes back to that equitable process too of if you give them software tools to use that are free, they can download them, they can practice on them, and they are the same one that are used in many places. And it makes it more accessible to the students to get a command of that material. And so I'm screeching into a halt. My takeaways really are about applying what I learned in my research experience in my pedagogical world in such a way that I hope benefits the students, that increases them feeling confident about their skills when they leave their undergraduate career. And part of that confidence comes from them feeling competent, which is, to me, related to skill sets, them learning the skill sets, but learning it not in a sit and take notes way, but in an actual doing way. And again, I'm not reinventing something here. I know that there's lots of experiential learning at U of T. I'm advocating certainly that there should always be more, and I think that students would advocate for that as well. One of the recipes being no assessments. I don't know how that would work throughout the undergraduate curriculum. That's another talk altogether. But so takeaways really revolve around things like the issues of competence. What defines competence? What makes a student competent when they finish? How much of that is about producing technical skills so they can be ready for graduate school, or God forbid, they go right into the workforce. You don't have to go to graduate school or medical school. Students want hands-on learning. They want to be able to make mistakes, and they want it to apply to the real world. They don't want to learn anything theoretical anymore. Like, it's okay. How many times I have this conversation with my high school grade 12-year-old all the time when he's in math. Mom, what do you need calculus for? I actually can't answer that. I don't know. I don't think you need it, actually. But when he does his data management class, which is stats, I'm like, dude, you'll use that all the time. And apologies for the mathematicians, but I don't know what you use calculus for. I know it's important, but they would love to be able to be given problems that make sense with the world that they live in right now. And so it's the intersection of all of this that I think becomes really important, and why it all becomes quite valuable to the students in the end is not one of these things, but how they're all put together. And that's everything. So thank you. And we have like 3 minutes, right?
Thank you, Professor Dockstader.
But I'll stick around for the people in the room as long as they want. So maybe you want to start with the people online. No? Up to you.
Yeah, I just want to say thank you very much.
You're welcome.
And I'm very intrigued by your research work.
Thank you.
And everything that you have done. But what actually got me more is the lab boot camp.
Yeah.
I mean, that is really experiential learning.
Yeah.
And I think every faculty in the university should be doing that.
Yeah.
It's a real challenge posing with our undergrads going out into the world for their, seeking a job or grad school. And and you
I agree. It would be amazing if they could.
They could. So before further ado, let's start with the questions. You think we should do online?
Only because I'll hang around, and the online people might.
Let's take from the audience first, and then we will interchange, right?
Okay.
So let's go from the floor first. Oh, do I use this one?
Or if there's no questions, that'll be super easy.
Oh.
Anyone have any questions? Go ahead.
Do you have any feedback from the students about if they didn't pursue a career in medicine or research, if they got the same kind of benefit from the boot camp that
Absolutely.
you saw? Okay.
Yeah, okay. So I've never asked that question specifically, so I guess I don't know. But what I would suggest is that what I found, so out of boot camp for students, what it does is it either says, now they know they like research and they want to continue in it, or occasionally they're like, I really enjoyed it, it was really super social, I love all my new people, but it's not for me. And I don't care at all, because that's a really valuable contribution to that, helping that person figure out their path. So that's okay. I think a lot of students at the end will say that they thought they wanted to go into research, and now they know that they do, or they're really interested in combining research and medicine. But I had more students who would say it gave them the confidence to go out and get that summer job interview. I had so many people say, I would never have applied for that job if I didn't have, if I hadn't gone through boot camp. So I would say more than anything that I'm aware of, the feedback I get from students is it really facilitates either fourth-year student projects. Now they have experience and they know what they want to go into for that, and or RA positions. And there are a few people who really, it leveled up their confidence in themselves more than anything else. I wouldn't say there's necessarily a difference in whether they were interested more in med school or grad school afterwards. I think that it probably was reinforcing for almost everybody, and probably 90% of them in a positive way, to go, yes, this is what I like. And then some people just said, you know what, it's not for me. And that's okay.
Mind if I ask one more?
Sure.
Just maybe a broader one. I'm just curious if you can, looking back on your own time as an undergrad and then maybe now on the faculty,
Yep.
do you, it was interesting to me that you said the students come up and they're so terrified is kind of the impression I got, that they're no confidence and they're
Yeah.
under stress.
Yes.
And you would think, being in a place like U of T,
Yes.
the world opens up for you, literally and figuratively. So I'm just curious of your opinion on why, is it the way that is it a pressure thing that students are under, or is it?
Wow. That's a huge question.
Yeah, I know it's, as you said, another talk maybe, but yeah.
That's more than a talk. That's like several theses. But, okay.
And was it the same? And do you notice a big difference from when you were an undergrad, I guess?
Okay, so when I was an undergrad, I had fun. When I ask my students now, do you have fun? They're like, no. I said, did you not have fun in first year or frosh weekend? No. So the difference is I had fun, and I don't think people have fun anymore. I'm also asking students who experienced COVID. Like even my own 23-year-old had first and second year during COVID. So it's changed so many things. But I would imagine if I asked the people before COVID at U of T, they might have still said no, they didn't have fun. I did notice so here's one thing that we all know. What's it called? The Dunning-Kruger effect? That smart people, and every student who goes to U of T, doesn't matter what they take, they're all smart. Every single one of them. Every single one of them. Because of the crazy marks they need to be able to get in here for any program. So everyone's smart. That's just done. And smart people tend to actually be really hard on themselves and tend to think, I don't know anything. And so I think, when I went to grad school at U of T, when I was an undergrad, I had fun. And the other thing is I did well. I got A's, I did well. But I was also a mature student. I was ready to go. I knew what I wanted. I worked hard. But I did really well. And people who go to grad school have also done really well. And so you still fit in that top 10, 15% where you can kind of feel good about yourself. And then you go to grad school, and everybody's as smart as you. And everyone did just as well as you. And in fact, this guy got way more awards than I did. And why am I here, right, with all these people? And the other thing is the more you learn, and really learn, not memorize, but learn, the more you realize you don't know something, right? The more you realize that there's so much more information and knowledge that we don't have. And so I think that is one piece of it. There's a whole bunch of smart people here who realize that there's so much to learn and everybody else is smart, and that can be really hard. And then there's the whole thing that we could talk about forever about the difficulties of being a student now, of which I don't know because I'm 54 and I didn't have the same experience that younger people would have had, so I can't speak to their experience. One thing I did want to say is I have so many students tell me it's too big. It's so big that they feel like they're a number. And so I do some first year presentations, and I always say, you have to find your people, right? You have to figure out who your people are and find your people, because there's like 80,000 students, 60,000-ish here at St. George. And so it's so easy for no one to talk to you and you not to be part of anything, and for all the commuters and that. And so I think you have to actively really try to find friends and colleagues and people to hang out with, because it's so big. I think if you don't actively do that, it won't happen. Because it's not like a small place where someone's like, hey, new person, come on over here. So that's what I hear. And I see some head nodding from younger people, so maybe that's right to a degree. So one of the best parts of boot camp, whether they're like, oh, this is for me, there's love in the room. And I genuinely mean that. Like the students love each other. They work together, they have fun. By the end of it, they're all going out together, they stay connected to each other. And so collaborating is absolutely the best thing about it. Putting them in small groups of varying skills, taking away the marks, so taking away the pressure, and then pairing them, mentoring them with someone who's young enough that they can identify with, but further on in their career so they really can guide the scientific process. I don't know how we figured it out right the first time, but we just did. Yeah. So we made it small.
Okay, I'll take the other one online.
All right, so we start first with comments. There is Tony Blair. He said, great presentation. Thank you so much.
Thank you.
And there is another one. This is from Jade. Thank you so much for this seminar. Great to hear from you again, Dr. Dockstader.
Is that Jade Huang?
Jade, if you are Jade Huang, please say yes. Probably, yeah. There is somebody, Stephanie, said, I graduated a while back. What research options are available to me? I have been reaching out, but this lecture sparked something.
Okay, well, that's the first thing then, I guess, right? Is having that motivation and that finding that confidence. So you have to go back and look at your experience. And I mean, again, it can be a whole conversation, but I'll try and keep it really short. In terms of school experience, you have to go back and you have to think, okay, what did I do? And what do I still feel confident in? So that's the other thing. If you feel like I have no idea how to pipette anymore, or I have no idea what stats are anymore, one, just don't put them on your resume. Or two, find an opportunity where you can go back somehow and try and practice those skills if you wish to. There are lots of opportunities to kind of rejig your memory online. I hate to agree with my 17-year-old who says how much you can learn from YouTube, but sometimes it's true. I wouldn't replace a degree with it, Hayden, but it can be true for certain. Like I said, the softwares that we use are freely available and downloadable. So if you've graduated a while ago, some of those actual technical hands-on skills, you might need to figure out a way to revisit. In terms of transferable skills, U of T produces people with the best transferable skills in the world, seriously. And there's a huge number of U of T graduates. We were like the third most hireable university in the world. I know they tell us a lot of these, so I can't keep track of them all, but I think we're like third most hireable in the world. And what that means is not that we're all experts in something by the end, but it means that, holy moly, you're a good problem solver, you're a great troubleshooter, you're a hard worker, and you're smart. And those qualities are really the most important for anything that you end up doing. So remember that you're hardworking, smart. What are the other things I said? I have a bad memory. Great troubleshooter, problem solver. Remember that you have all of those skills already and you use those all the time. So try and find yourself some confidence in the skills and abilities that you already have. And go ahead. Yes, Jade, hi. Oh, I think I owe her a reference letter. That's why she's here. Either that or I just did it. Okay, go ahead. Yeah.
How would you differentiate the value between the confidence and tied to the competency that shows up in your boot camp versus the confidence that would be obtained by competence that's determined outside of what you would learn yourself? So like a certification exam that determines you're competent in something, and the confidence tied to that.
I think I go back to the ability to be allowed to make mistakes. Making mistakes we say this to our kids all the time, mistakes are how you learn, but then you come to the classroom, or even for a certification, you're not going to be certified. You don't mean certification for boot camp, you mean like certification for...
No, like I'm an MRT, so I had to go through a certification exam to determine that I was competent to be able to practice.
Yes.
So I'm just kind of wondering what the difference would be value-wise between the two.
It's a great question. It's really for the boot campers, competence was not enough for them to feel like they could go out and get a job because they're competent, because they're smart, they're troubleshooters, they're problem solvers. But they didn't have the confidence to allow them to go do that. And so competence is a way to say, aha, tick the box. But it was the confidence building that came from what comes beyond ticking the box, which is all the stuff you don't get to see before you tick the box, like making all the mistakes, but understanding why you made the mistakes and collaborating with others. Like, it's all the gray area, actually, that leads to you to be able to do something, but to be able to do it in a way that you feel like you can do this. You can go for that interview. Yes, I do know how to have that skill. Yeah, definitely. That's a great way to say it. So the person in the audience said, would I say it was more personalized? And I would agree with that, for sure.
Okay, thank you. I have another question here from Huda.
Yeah, that's from me. First of all, thank you so much. Fascinating research and work with students. My question is is there any baby steps to enhance this or to make it get into how the labs are run in general?
It's a discussion we have. It's really hard how to figure it out practically. So I'll tell you, and I don't know where this goes, but I haven't even asked my boss what she thinks of it, but here we go. What I would love is if we had a boot camp that was for credit, but it was credit, no credit. So that way, people don't have to try and fit it in. Like, we're literally trying to just squeeze it in between the end of exams and the start of new courses. It's not equitable because people have to work, people have to go back home, what have you. So if it were during the year, and it could be worth credit, then people could take it. But then we lose what I think makes the boot camp what it is. And so in order for it to be credit, it has to be just credit, no credit. There's no marks. You come, you do the work, you will get your credit. End of. So that's the way I think would make it most accessible to other people and bring it in. Now, still, I don't see how we could do that for 2 weeks because they're taking their other courses. So it would change that. But there doesn't have to be only one way to do it. I mean, we can try all these things and see what happens. Some will be more fruitful than others. We got really lucky the first time, but there's lots of limitations on the approach that we have right now too. So maybe to bring it to more people, we'd have to have something that just wouldn't be 2 weeks in a row. Yeah.
Thank you. It seems the discussion can continue on, but yet we'll be ending it here for tonight. And for closing remarks, I'll call on my colleague, Dr. Natalie Hoffman.
On behalf of Woodsworth College and the Alumni Association, we'd like to thank you very much for talking with us tonight. We really enjoyed this discussion. I would also like to thank all of our guests here tonight for joining in person and online. If this is your first time at one of our events, we hope you will join again. And please keep an eye out on your email for other information regarding Alumni Reunion Week, and we hope to see you at our future events. I will also take this opportunity to express our thanks to Carolyn Peralta, the Alumni and Development Officer at Woodsworth, for her steadfast support in making all of our events successful. We also extend our gratitude to Woodsworth College for their continuous support of our alumni board events. Also, thanks to the U of T Campus Media Productions for their technical support tonight. With the 50th year anniversary celebrations, Woodsworth College continues its unwavering dedication to providing exceptional student experiences. There are various ways to show your support, including by donating to the college. For more information, there are QR codes around the room as well as shared online, so please visit that link and feel free to talk to any of the alumni board members for further details. Before saying goodbye, I would like to mention we have a short survey for you. For our online audience, it will pop up once you leave the Zoom session, and we'll also be sending it in the coming days in your inbox. It would help us immensely if you could take a minute to fill it out. We would love to hear from you so we can better tailor our future events. That concludes our evening. Thanks, everyone, and good night to our online audience. For everyone here, please join us for some refreshments and drinks in the background. And please stay to mingle for a bit. Thank you very much.